Hello friends, again welcome to my channel. Today we are going to discuss about one of the important metabolic pathway, that is the biosynthetic pathway, within which there is the synthesis of glycogen. This glycogen, which is considered as the major storage form of the carbohydrate. It is the storage form of carbohydrate within the animal body, similar to that of the starch, which is present as a storage molecule within the plants. Now, the glycogen molecule, it is considered to be a homopolymer. It is an homopolymer, means it is made up of the same type of the monomer units, that is the glucose monomers. Its monomer unit, it is the glucose. So it is made up of the repeated units of the alpha D glucose, and these glucose monomers, these are linked with each other by the alpha 1 for glycosidic linkage, which is a link between the first carbon atom of the active glucose residue to the sixth carbon of the approaching glucose molecule. Again, the glycogen, it is a complex structure. No doubt it is a homopolymer, but it possesses the complex structure. So the branched fragments, these are present within the glycogen molecule. Once there is the chain consisting of 8 to 10 glycosidic residue in the glycogen fragment, then at that time the branching it is get begins by the alpha 1 4 glycosidic linkage. Means within the linear form there is the presence of the alpha 1 4 glycosidic linkage to join the glucose residues, and within the branches there is the presence of the alpha 1 6 glycosidic linkage. Now, the glycogen, it is preferentially gets stored within the liver and the skeleton muscles. Within the liver, it is used for the maintenance of the blood glucose levels. Means whenever within the blood there is the higher concentration of glucose it is present, then this higher concentration of glucose it is get converted within the glycogen, and it is get stored within the liver and skeleton muscles. Within the muscles, this glycogen it is utilized to meet the energy needs of the muscles. Means after the exertion, again this stored form of the after the exercise this stored form of the glycogen it is get converted within the glucose. So it is within the muscles, this glycogen it is used for the to getting the energy. Now the glycogenesis, it is the process through which there is the synthesis of glucose, in which the glucose molecules these are added to the glycogen chain for the storage purpose, for the purpose of storage of the glucose molecule in the form of glycogen. Means in other words we can say that the glucose, higher amount of the glucose it is get stored within our body in the form of glycogen through the process of glycogenesis. This glycogenesis process it takes place in the cytoplasm of the cells, in the muscle, liver and the adipose tissue. The substrate, that is the chemical substance which is utilized to initiate the glycogen synthesis, it is the UDP glucose, that is the uridine diphosphate glucose molecule. It is acts as a substrate, and then it is get converted within the glycogen molecule. Here it is the structure of the glycogen molecule, within which the glucose monomers these are get linked. Within this linked form you can observe that within the linear form there is the presence of alpha 1 for linkage, and whenever the branching it is carried, only at that branching point there is the presence of alpha 1 6 linkage within the glucose monomer. In the linear form only there is presence of alpha 1 4 linkage, and only at the branching site there is the presence of alpha 1 6 linkage. Now we are switch over to the different steps which are involved within the glycogenesis. So firstly, for the conversion of the glucose into the glycogen, the first step within the EMP pathway it is carried out, that is firstly there is the phosphorylation. For the purpose of activation of the glucose, there is the conversion of the glucose into the glucose 6 phosphate. There is conversion of glucose into the glucose 6 phosphate by the activity of enzyme, the glucokinase or hexokinase. That is this kinase enzyme, it is responsible for the addition of phosphate group to the sixth carbon of the glucose molecule, and there is the conversion of glucose into the glucose 6 phosphate. And within that step, there is the utilization of one molecule of ATP. ATP, it is responsible to provide the phosphate group for the phosphorylation of the glucose. Now, the glucose 6 phosphate, again it is get converted within the glucose 1 phosphate. Means the phosphate group which is present at the, which is attached to the 6 carbon of the glucose molecule, it is get transported within the first carbon by the action of enzyme phosphoglucomutase. This reaction, it is passing out through the obligatory intermediate product, that is the glucose 1,6 bisphosphate. Within the third step, this formed a glucose 1 phosphate by the activity of phosphoglucomutase enzyme. It is get converted within the UDP glucose by the action of the enzyme UDP glucose phosphorylase. UDP glucose phosphorylase. Now this UDP glucose phosphorylase, it is responsible for the addition of the UDP molecule to the glucose monomer, and along with that there is the removal of phosphate group from the glucose molecule. The pyrophosphate which is formed, it is then later hydrolyzed by the pyrophosphatase into the 2-phosphate molecule. Now, after this step, whenever there is the conversion of glucose 1 phosphate to the UDP glucose, which is a substrate to initiate a glycogen synthesis, now here there is the requirement of the enzyme glycogenin. This glycogenin enzyme, it is important for the initial synthesis of, or the for the initiation of glycogen chain synthesis, for the initiation of glycogen chain synthesis. And then this chain, it is get enlarged or get elongated and branched by the other enzymes which are involved within the glycogenesis. But for the initiation of glycogen chain synthesis, there is the requirement of this glycogenin enzyme. Now this glycogenin enzyme, it is an homodimer, and preferentially it possesses a tyrosine residue on each of its subunit. As it is homodimer, it means it made up of the two same type of the subunit. On each of this subunit there is the presence of a tyrosine residue which serves as an anchor for the reducing end of glycogen, which acts as an anchor for the reducing end of glycogen. That means to the reducing end at which the end of the glucose monomer to which there is the addition of the glucose, another molecule of the glucose, it is called as the reducing end. It is said to be the reducing end. Now this glycogen molecule or the tyrosine residue present within the glycogen molecule, it is responsible for the removal of UDP molecule which is attached to the glucose molecule, and it is responsible to joins the multiple glucose monomers to each other by the alpha 1,4 glycosidic linkage. By alpha 1,4 glycosidic linkage means each of the synthesized UDP glucose molecules, these are linked with each other by the activity of glycogen enzyme by the removal of the UDP molecule by forming alpha 1,4 glycosidic linkage, and majorly this process, it is continue up to the addition of 8 to 10 glucose monomers. Once the chain of the 8 glucose monomers, it is get formed by the activity of glycogenin, then there is the activity of glycogen synthase enzyme. This glycogen synthase enzyme, it binds to the growing glycogen chain and adds the UDP glucose to the 4th hydroxyl group of glucosyl residue. 4th carboxyl group of the glucosyl residue on the non-reducing end of glycogen chain. Non-reducing chain of glycogen chain, forming the more alpha 1,4 bonds within the process. Means to the non-reducing ends of the glucose molecule, there is the addition of the glucose monomer by the activity of glycogen synthase enzyme. Now as we said that majorly it is the homopolymer and it is made up of the glucose monomers, but this is a branched homopolymer, so the branching it is present. So for the branches which are present within the glycogen molecules, these are made by the glycogen branching enzyme. Glycogen branching enzyme, which is also known as amylo alpha 1,4 alpha 1,6 transglycosylase. Amylo alpha 1,4 alpha 1,6 transglycosylase, which is responsible for transferring the end of the chain onto the earlier part. The end of the chain onto an earlier part via the alpha 1,4 glycosidic linkage. Alpha 1,4 glycosidic linkage and responsible to form the branches. Responsible to form the branches which further grow by addition of more alpha 1,4 glycosidic unit. Means only at the branching point there is the presence of alpha 1,6 linkage at which there is the activity of this transglycosylase enzyme. Now within the synthesis of glycogen molecule, only the 1 ATP it is required as per glucose molecule which is incorporated into the polymeric branch structure of glycogen. Only at the first initial step, whenever there is the conversion of glucose to the glucose 6-phosphate, at that point there is the utilization of 1 UTP molecule for the purpose of phosphorylation of the glucose or for the activation of glucose molecule. Now the important enzymes which are involved within the glycogen synthesis, it is the glycogen synthase which adds the glucose units to the non-reducing ends of existing chains, and these chains, the initiation it is carried out by the glycogenin enzyme. Then the glucosyl 4,6 transferase. This glucosyl 4,6 transferase, it transfers the 7 glucose residue long pieces from the non-reducing ends of the chains to create the internal branches. To create internal branches with the alpha 1,6 glycosidic linkage. Here it is the structure of or the pathway through which we can observe how there is the synthesis of glycogen. Firstly, the glucose it is converted within the glucose 6-phosphate. Then from the glucose 6-phosphate there is the conversion into the glucose 1-phosphate by the phosphoglucomutase, which is an reversible reaction. Reversible reaction. Then there is the utilization of 1 UTP molecule. This UTP molecule it is get converted within the UDP, and there is the that is UDP glucose phosphorylase. That is the phosphate group from the UTP molecule it is get removed. It is get converted in the UDP and it is attached or condensed with the glucose molecule, and there is the formation of UDP glucose. UDP glucose. Now after this UDP glucose which is acts as a substrate, then the glycogenin enzyme it is responsible for the synthesis of or for the to initiate the glycogen chain synthesis. Glycogen chain synthesis, and it binds the UDP glucose molecules. Whenever there is the 8 UDP glucose molecules, these are joined with each other by the alpha 1,4 glycosidic linkage. Then there is the role of glycogen synthase enzyme. This glycogen synthase enzyme it is responsible to continuously adds the glucose monomers to the non-reducing ends of the glycogen chain. Non-reducing ends of the glycogen chains present on the glucose monomers, and by this way there is the synthesis of glycogen. Within the synthesis of glycogen from the glucose, only one molecule of ATP it is get utilized. So here it is the all over process of glycogen synthesis. If you like this video, please subscribe my channel and also share this video with your friends. Thank you.